Hello, so my name is Maddie, and today I'll be presenting on HIV-1 and AIDS, and the treatment breakthroughs we have had in the last couple of decades. I'll be specifically highlighting the potential uses of CRISPR-Cas9 technology, as well as going through other treatment strategies that have been useful to us so far. I will also be touching on the history of the AIDS epidemic and breaking down how the virus operates inside the body. So I hope you enjoyed the presentation. I also really quickly want to just point out that in the bottom right here is Princess Diana shaking hands with an AIDS patient. So the reason Lady Di is shaking the AIDS patient's hand was a big deal, was because people didn't understand very much about AIDS back when it was first discovered, which was during that time. It was the late 80s. It was originally considered a gay man's disease because that's who, the population it was first discovered in. People thought that you could contract it just by standing next to somebody who had it, or by shaking hands with somebody, or kissing them, or sharing drinks. So there was a lot of panic. And Lady Di made a statement by actually treating somebody with respect who was dying of AIDS and shaking their hand like a normal person. It was a really big deal, and it's a very iconic picture, and I just really liked it. So. But anyway, it was first discovered in 1981. It had probably been around by around maybe the early to mid 70s. And like I said, it was the first few AIDS patients were gay men who lived a rather promiscuous lifestyle and had several partners. And this was before that it was known that there was this basically like silent virus that was on the loose. And yeah, so people started dying of it, and doctors were absolutely baffled. It was just like people's immune systems just started failing, and they had no idea why. And then people who had received blood transfusions during that time also started coming down with a similar condition, where there were strange kinds of pneumonia that were happening, like just really rare strains of diseases. A fungus, like a brain fungus, was common. And these nowadays, those are considered AIDS defining factors. The signs that AIDS is really, aside from the low white blood cell count, those are some of the key indicators that AIDS has been triggered, or it's set in. Anyway, we've come a really long way since then. If you want to look at that, there's a brief and as far as recent research and recent breakthroughs are concerned, it's pretty much prior to everything I'm going to be talking about. So there's that history. So what is AIDS? What is HIV? HIV stands for human immunodeficiency virus. There are two types. There's type 1 and type 2. Type 1 is the strain that you typically, or the strain. There are several strains off of that, so I don't know. But HIV-1 is what you see in the general population, whereas around in North America and Europe and pretty much everywhere except West Africa. HIV-2 is almost exclusive to West Africa, where almost 50% of the population is infected. It's been tragic and catastrophic, but it's also, their strain is less infective. However, it progresses more quickly than HIV-1. HIV-1 is very infective, but especially with antiretroviral medication, it can be slowed significantly. Anyway, so HIV is a retrovirus, which is a certain kind of virus that uses reverse transcription to copy its DNA within the cell. It specifically targets white blood cells, namely your CD4 plus cells, aka the helper T cells, as well as the macrophage cells, which are basically like the police system of the white blood cells of your immune system. They present pathogens to the helper T cells, as well as like dendritic cells. I think I know they're involved. I'm not sure if they present it to the dendritic cells, but anyway, they present them to the helper T cells, and from there they are consumed and destroyed. Those are the two main types of cells that HIV targets, as well as the CDC5 cells, which are kind of similar but not quite the same. And that's typically either it targets both or it targets one, the CD4 plus cells being the most common. Anyway, and then so over time it weakens the immune system and eventually causes AIDS, which is acquired immune deficiency syndrome. So AIDS is the final stage of HIV infection. It is where the immune system is so critically damaged from reactivation of HIV that it is basically susceptible to all kinds of illnesses. One of the common things that you hear is like with COVID. I was hearing on the news that a lot of HIV and AIDS patients were really concerned because they don't have the police system in their white blood cells anymore. And not only that, they don't even have the fighter T cells, the helper T cells that are actually clearing the illnesses. So they're susceptible to almost everything. It towards the end, it's when the white blood cells drop even further below the critical point, which is 200 parts per million. It can be catastrophic, and it's a lot of strange, like a very rare kind of pneumonia is actually one of the markers that AIDS has finally set in HIV patients. So yeah, opportunistic or secondary infections eventually will cause death. There is no known cure, but it can be suppressed with regimented antiretroviral drug therapies. I'll get into that on the next couple slides. And then here I just have a couple of statistics. So there are 38 million people currently living with HIV and AIDS. There are 79 in America, in the US. 79.3 million total people are infected globally. Have been infected globally. 38 million people globally, worldwide, are infected right now. And then 79.3 million people, it is estimated, have been infected since AIDS was first reported. There are a lot of deaths and a lot of other illnesses that were probably attributed to countless people who actually had HIV, particularly in the first 2 decades. But that being said, 36.3 million people are estimated to have died. It's actually between, the estimation is between 30 and 45 million. This was the average that they came up with because, like I said, there are a lot of unreported deaths as well because of the secondary infections being the cause of death. So, HIV, it was originally thought that the way that it interacted with cells, so the way that, I'm sure in biology class you've heard about viral spikes or about ways that the virus actually binds to the cell or tricks the cell into accepting it, because as you know, the protein receptors are very picky. The HIV virion, so here's my fist, right? The size of an HIV virion is actually probably more akin to my skin cell. So imagine, here's my whole fist, this is your regular cell, this is a white blood cell. And the size of one of my skin cells is probably the size of an HIV virion. They're very, very small. This video right here, which I'll play for you in a minute, is one of the first that microbiologists and virologists were actually able to isolate. They were to be able to zoom in and dilute it enough to be able to actually see what was going on. At first, they thought that there was a viral spike, not just little. Turns out there are 3 little, tiny little, needle-like tendrils that come out. The virus is normally closed, and so what happens is it opens to reveal the viral spikes or to reveal its genetic material when it binds to the cell. Originally, virologists thought that it was just this giant spike, basically, because it's just, I have no idea why, but that's what they originally thought. So the virion opens to fuse with the host cell plasma membrane. Let's see, and then so it targets susceptible CD4 plus T cells and macrophage cells, like I was discussing earlier. So here's the video. It's actually really cool. It's slowed down significantly. Do you see the 2 little spikes? This is actually a really cool video because do you see it opening and closing? It almost looks like a sea anemone or something. And actually, so when it's open, that's one of the only times that it's susceptible to even antiretroviral drugs. When it's closed, it's almost like a cocoon. It's almost like it's first of all, it's wrapped in the host's own cell membrane. So it takes parts of the cell membrane and basically cocoons itself in that, and that helps with camouflage and avoiding white blood cells and other threats, I guess, or detection basically. And then so only when it opens is it even susceptible to antiretroviral drugs. It's one of its weakest moments, but this is slowed down significantly. I mean, it's taken a very long time for them to even isolate this moment. Anyway, so that's actually one of the targets of potential AIDS treatment. Mostly it's a preventative thought, I believe, but yeah, that's just one of the targets that they're thinking about. So let's see. there we go. Two little tendrils. One. Let's go. So this is the life cycle of HIV. There are 3 stages of HIV infection. The first one being infection. So the virus enters the bloodstream through whichever avenue it did, and interacts with the main binding sites of CCR5 and CXCR4 plus cells, or CCR5 and CXCR4. Binding sites on susceptible macrophage and CD4 plus cells. So the CCR5 and the CXCR4 are the binding sites, not the cells themselves. Like I was discussing earlier, the macrophage and the CD4 plus T cells are the main targets. So what happens is it binds to those receptor sites. Let me see if you can see my mouse. I don't know if you can or not. Here. So it binds to the receptor sites right here. And then so from there, it injects its viral DNA, where the viral DNA is through reverse transcription, it transcribes it becomes part of our own DNA. It replicates by converting its RNA into double-stranded DNA. So this is actually where the latent viral reservoirs are established. It's during this time. So through integration with the host cell DNA, it becomes proviral DNA. Where it's then also broken down into viral DNA, and the viral proteins, again utilizing our own cell processes, are assembled, and then they bud out of the cell. So like they don't actually, the thing that what I was taught like on our first day of biology 222 was that typically when viruses leave the cell, they destroy the cell, or they often it creates cell destruction. Originally, with HIV, that doesn't happen right away. It leaves the cell intact. It buds vertically through the top of the cell, and it leaves the cell functioning completely normally. What it's officially done is actually created more viral virions to infect further cells, but then also it has like I said integrated itself into the host DNA, or it's your chromosomes. It's actually integrated itself into your chromosomes, so there's almost like an imprint of the viral DNA, of the viral RNA embedded into your own chromosomes. And that's where the provirus comes from. It continues to replicate kind of quietly in the background, which leads us to part 2. All right, so that leads us to stage 2, which is the latency stage. So the latency stage is when there are still alive HIV provirus reservoirs, which are basically viruses that are persisting within the host's cellular DNA and their genome, with the chromosomes, like I was talking about. So they're still basically just hanging out unobtrusively, kind of in the white blood cells. And what happens essentially is that these reservoirs are, like I said, unobtrusively replicating in the background, but they're also, the provirus is able to spread to adjacent cells. So other cells, susceptible CD4 plus cells or macrophage cells that are interacting with that cell, also become infected by proviral virions or proviruses. So the proviruses create new viral reservoirs inside these cells. And so basically it's gradually, exponentially, but also still gradually becoming more and more of an issue. It's quietly replicating in the background. I'll get into how antiretroviral medications work, but we are able to control the viral load that's present in the blood. But the main problem with treating HIV are these viral reservoirs that are still persisting no matter what we do. They're really hard to track down. No one knows how to like tease them out of the cell. I mean, it's part of the DNA in these cells. And so when it's been replicating this for this long, there are so many cells that are infected. It's almost like I guess the way that I understand it, I would like almost attribute it to like treating cancer, you know, where all kinds of other cells are going to be damaged in the process of trying to weed out these certain cells. So I'll get to that in a minute because that's the key to CRISPR-Cas9. But anyway, so reactivation occurs when, so reactivation means when these latent viral reservoirs begin actually producing live virion again. Not just sort of like, being all sneaky and hiding in the background and like just replicating with your own DNA and blah, 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 all that nasty stuff. It's when they're actually, when the viral loads become detectable again and they actually begin to override the white blood cells. Or when the viral reservoirs have, like, basically it's when, like, so some antiretroviral medications can trigger reactivation as well as just immune detection. So when it's detected, the virus reactivates, and that's when AIDS begins to set in. So destruction is when the virus dominates and thwarts the limited immune response. So the proviral reservoirs and sort of, like I said, so AIDS, okay, let me backtrack and slow down a minute. So HIV takes years to develop into AIDS, even longer, decades, if you are, if the infected person is judiciously and well, diligently, I guess I should say, taking their antiretroviral medication. So every day at the same time, every day, it's a constant onslaught, and it helps, it keeps it undetectable. It's not transmissible through sex, anything like that. Eventually, however, no, I would think that the person who's basically been survived, or that I know of that survived the longest with AIDS or HIV, has been Magic Johnson, who has had it for, I think, 30 years now, 40 years even, and he's still thriving. He takes his medicine all the time, every day. Granted, he has access to some of the best medical care in the world, but it's unknown how long the antiretroviral medication can suppress the HIV from turning into AIDS, but it's quite some time. So eventually, however, the white blood cell count will drop below a critical number because the virus has ultimately destroyed and corrupted most of the immune system. So there are no more. The CD4 plus count has dropped below 200 parts per million, which is like critical levels. It's gotten pretty bad, and the macrophage cells, which are the ones presenting the pathogens, have also been corrupted, as in they're presenting. They're not doing their job either. They're basically destroyed. So the immune system has been destroyed, and then that leads to stage 3, which is AIDS. So the HIV provirus has infected and destroyed most of the CD4 plus T cells, and the crippled immune system has become susceptible to frequent and opportunistic infections, which is what eventually causes death. It's not the virus itself, it's what it does. So let's see. All right, so these are the current HIV therapies and harm reduction strategies. I want to start at the bottom of the slide because it just reiterates everything I've said. We still do not have a cure. What I'm going to be talking about in the next slide, or the next 2 slides, is a potential research or a research cure that we have, that we've been doing or we as human beings, I guess, have been doing clinical trials on. But the best treatment strategy we currently have is prevention. The best way we can treat HIV or stop its spread is by not getting it in the first place. So, however, so then I'll start on the left, or the right actually. So PrEP and PEP. So on that note, PrEP is pre-exposure prophylaxis, which is basically pre-exposure prevention. It's intended for those with ongoing risk of HIV infection, for instance, at-risk populations, such as those who, let's see, drug addicts, people who have an HIV-infected partner, gay men in particular, and then also certain socio-economic groups and ethnic groups are more at risk than others. And they are highly encouraged to be taking this. I think most insurance actually covers it for free. But anyway, it significantly reduces the likelihood of contracting HIV through unsafe sex and sharing needles, or basically just being exposed to it. So if there's a chance that you may be exposed to it on a regular basis, if you take PrEP every single day, they've actually narrowed it down to, I think, 1 pill. It's called Truvada. if you take Truvada once a day, even if your partner, you know, or you are aware your partner is HIV positive, and even if they're on antiretroviral drugs, if you're taking PrEP, your chances of contracting HIV have dropped from, I don't know what it's dropped from, but it's about 97% chance you probably won't get it. PEP, on the other hand, is it's kind of like, I don't know if you've ever heard of Plan B. It's an emergency contraceptive pill for pregnancy. PEP is basically the HIV version of that. It's only to be used for emergencies, or when you have within 72. And so the bottom line here is within 72 hours of suspected HIV exposure. So let's say you're a healthcare worker and you got stuck by a needle, or you came into contact with a patient whose blood you are aware of was infected with HIV. PEP is post-exposure prevention, and yeah, the sooner you take it, the better. But that can also prevent HIV infection, even if you've actually been exposed to the virus for sure. So then the other main point, and this is if someone were to become HIV positive, our current form of therapy is antiretroviral drug therapy. So CART is combined antiretroviral therapy. It inhibits viral replication mechanisms through several key points in the viral replication process, which is on the next screen, so just hang on. But it keeps viral load virtually undetectable and untransmissible through sex. So when your viral load is undetectable, it's not likely to be spread through your bodily fluid if it's not detectable in your bodily fluid. So that is lowered to like 98% chance again of not transmitting it. The drawbacks is that it cannot eliminate the latent viral reservoirs that I was talking about previously. Therefore, it cannot cure HIV. What it does is it buys you time, and it also helps you not to infect other people, and it improves your quality of life significantly, to a point. The other negative is that there are a lot of side effects. Long-term use can lead to neurological problems, bone marrow issues. Let's see. Several things just from extended use. It's also an expensive, daily regimented, so like I said, same time every single day, and it's a lifelong treatment. If you stop taking it unexpectedly, well, sometimes actually doctors do have you not take it anymore, because that can actually like kind of almost like reshuffle the HIV mutations, because the other thing that makes HIV so hard to treat is that it mutates very quickly, it replicates very quickly, and it mutates very quickly as a result. And so that being the case, a lot of the time, the surviving virion in the bloodstream will become immune to the antiretroviral drugs that they've been introduced to. So the surviving ones are the ones that didn't get killed by the drug. So sometimes doctors will actually stop, especially if patients have been on the same drug cocktail for a long time. They'll actually stop treatment and then resume it, and they'll see the viral levels drop again. But yeah. So again, it's a daily, lifelong treatment, and there are a lot of very serious side effects. They're very intense drugs. So let's see. So yeah, so these are some of the targets of antiretroviral drugs. They target key points in the HIV DNA replication process, and then so they are the current treatment standard for HIV-1 treatment. Again, an undetectable viral load doesn't mean cured. Like with hepatitis C, I know that when it's an undetectable viral load, there aren't viral reservoirs that are kind of hanging out in the background, and it actually does basically mean cured. Your body's able to clear the virus. That is not the case for even with, like, for example, like HSV-1, so herpes. Herpes simplex also has latent viral reservoirs, and they kind of hang out in the background. They get reactivated, and they're really hard to track down because they've integrated themselves through reverse transcription. So anyway, these are some of the ways that antiretroviral drugs work. And then, let's see. And we're finally to CRISPR. Okay, so applications of CRISPR-Cas9 gene editing for HIV-1. So there are 4 main ways that my article discussed in which CRISPR-Cas9 can be useful in actually curing HIV, not just treating it, but curing it. The main ones I want to talk about are in white, but I'll just quickly go over the ones in black first. So the first one is just straight gene editing. particularly editing or mutating or silencing the protein binding sites, so the CCR5 and CXCR4 binding sites on the CD4 plus helper cells, in which case those would be mutated or silenced. And then also with gene editing, removing, so actually editing the host gene to remove the integrated viral genes from the host chromosomes. The other one is disabling critical host factors that HIV-1 relies upon to replicate, such as mutating the protein binding sites. But that would also include, it would basically essentially restrict viral activity within a cell. Again, both of those I feel like could have critical side effects, especially since CRISPR-Cas9 has not been researched to the extent that it's even on like patient or human trials yet, especially for a few of these. The one that is the farthest along is number 2. Which is cleaving the HIV-1 virus and editing the genome and RNA. So by cleaving, what it means is, so it's called the shock and kill strategy. So what it's doing is, so like I told you before, there are these latent viral reservoirs that are basically persisting in spite of everything we can throw at them. The shock and kill strategy is actually, so there are drugs, and also through there are drugs that can reactivate the HIV virus or HIV, and in doing so, it almost teases out those cells that are that have the proviral reservoirs inside of them, and it also so it reactivates the provirus to pinpoint the cells that are affected and annihilate them. The problem with this is, so this is under extensive research because it actually is very promising. However, the problem with it is that not all of the, it's not very precise. So there are cells that that end up being affected by this that shouldn't have been. It's almost, I would probably assimilate it more to chemotherapy than anything again. So there are others, it needs to become more precise, and there needs to be a more, it needs to be more accurately pinpointing of which cells to attack and which cells to edit. Let's see, the other one is forcing the expression of, well, let me move my face. I can't even see it. Hold on a second. My face is in the way. Oh, okay. Yeah, so sorry, my face is still in the way, but I remember what it was. So using CRISPR-Cas9 gene editing to express innate immunity, this would be done through actually stem cell research. So there have only been 3 people so far to ever have been fully cured of HIV. And there was one recently in the news, and it was a woman who actually had contracted it, and she just cleared the virus. She's one of 3 people ever to have been cured. And she had an innate immunity to it, which is extremely rare. And so when she had a baby afterwards, and the baby was, again, HIV negative, everything was cool. They wanted her placenta. It's actually it was like a scientific diamond, I guess, because HIV is also spread, I forgot to mention this earlier, but it's also spread from mother to child through the placenta, not just the birthing process, but through the placenta itself. And so her immunity, those cells that made her immune, and also could be cultivated into stem cell, or stem cells could be taken from it to study. It was basically, it was taken to a lab immediately the minute after her baby was born. It was like in a box and everything. I have the link at the very end of the presentation. It's kind of cool. But yeah, so she through stem cell research, they might be able to use CRISPR-Cas9 to, like with the bacteriophage viruses, or the bacteriophage in the bacteria, they could edit the genome to express innate immunity, and not just edit the virus out of the cells, but actually to help the body express immunity in and of itself through stem cell research. Okay. Okay, almost done here, I promise. So this is the last thing. I thought the coolest part of this entire project was actually the history of HIV, more than even the biology. So this is the HIV memorial quilt, and it commemorates 40 years of AIDS, and it has 50,000 plus panels and growing. What it is each panel is about the size of a coffin. It's about 6 feet by 3 feet, and it has several quilt pieces on it, and they all represent different people, or different people that have passed away from AIDS. Like I said, it honors over 105,000 individuals who have died from AIDS. It weighs more than 54 tons. And in 1996, the quilt covered the entire National Mall in front of the White House. So, that giant, like, 3 football field sized lawn in front of the White House? It covered the entire thing. And that was in 1996. And it actually only accounts for about 20% of the people who have passed away from AIDS. And just the people that knew about it and wanted to make a quilt. So like I said, about 20% of the people in the United States who have passed away from AIDS. So what would that make that? 2 million? No. Yeah, a little over 500,000 people total that are unaccounted for at this point. Total late. Yeah, okay, sorry. It's a little late. I've been doing finals all week, so. Anyway, but yeah, it only covers a fraction of the people that actually have passed away. So in conclusion, here it is covering the entire National Mall in front of the White House. Again, this was in 1996 when it could still do so. Now the quilt is in pieces and it's basically in storage, and you can actually order it into your community to have it brought in and shown if you wanted to do something like that. It's just the entire thing. It's hard to find a space big enough to actually display the entire quilt anymore. So like I said, here's 1996 in front of the White House. So there are further reading references I wanted to just bring up really quick. I know this is getting really long. I'm sorry. There's a book called And the Band Played On, Politics, People, and the AIDS Epidemic. It's by Randy Schultz. He actually ended up testing HIV positive in late 1987 after this book was published. And he passed away in 1991 or 1994, I forget. But about 6 years later. But anyway, it's a historical narrative that lends a voice to the beginning of the AIDS epidemic. My mom was the one who told me about it, and she said, and I was like, oh, it's not even like recent, it's not relevant. And she said, well, actually, no, it's almost, sometimes the beginning is the most important part. And anyway, it brings to life the first people, or the first statistics. They become statistics, they become nameless faces, but it brings them to life. It brings a voice to the first people that ever had it. And I really loved it. It was a really good book. Anyway, and so then there's the AIDS Memorial Quilt, which has a history. It also has additional resources and other like artistic performances about HIV. And then life science. So this is what I was talking about in the previous slide about the third person ever to be cured using HIV stem cell transplant from umbilical cord blood. Anyway, yay! I hope you really enjoyed my presentation. I really enjoyed giving it. I could probably talk for another hour if you wanted to hear my voice that long. But anyway, happy finals week, happy spring break, and thank you for listening. Have a great day. Bye, guys.